Hello, my name is Mr. Chipman. This is 4.6 regulation of the cell cycle. It's for AP Biology Unit 4. I teach AP Biology and just honors biology at Murray High School in Murray, Kentucky. This is the last section of Unit 4 for AP Biology. And if you're in my class, this is the next to last unit that we're going to do before Christmas break. We will complete Unit 5, but we are moving right along in this class. After this unit, we are halfway through as far as units are concerned. Maybe a little less than halfway as far as overall sections. All right, so here's the cell cycle. We're talking about regulation of the cell cycle. So the cell cycle, remember, it's made up of interphase, mitosis, cytokinesis. Interphase is G1S, G2, mitosis, prophase, metaphase, anaphase, telophase. All these phases are important, and these important phases need some regulation. So we're going to look at those kinds of regulation. Before we do that, before we get to the checkpoints, I want to connect this to cell communication because this is really important, and some of that regulatory function has to do with cell communication. Cells, one of the ways that cells know whether or not they should be dividing is if they are in contact with one another. Say, for instance, you get a paper cut on your finger, right? You get a paper cut, and those cells that were touching each other just a few seconds ago are no longer touching each other. And so there's a signal that says, hey, we need to repair this damage, right? And then they repair it. And then they stop repairing it once it's repaired. How do they stop? Why do they stop? Because they can sense one another. Cells don't have eyes, ears, and noses, but they can sense one another based on the proteins that are located on their cell membranes. And so they sense one another's presence, and that sensing, that detection, that communication, that reception, transduction, response, right, allows the cell to cease its cell cycle, or to slow it down, or to go back to normal as opposed to being in repair mode. And so that allowing them to, that direct contact allows those cells to divide properly. When an organism develops, the same kind of thing is used, right? Cell is able to sort of detect its surroundings and know when it should be dividing, when it should be specializing, which we're going to look at in later units, the ideas of cell specialization, how cell communication it directly involves cell specialization. But we'll look at that as we go. But I just wanted to touch base on that because I think the idea that cells when to divide and how often to do so is definitely tied to whether or not cells are in direct contact with one another, obviously in a multicellular organism. So let's look at this idea of cell checkpoints. There are 3 checkpoints found in the cell cycle. A checkpoint is exactly what it sounds like, right? It is a place where you're like, okay, are all these things present, right? If you've ever been through like a checkpoint while you're driving, you get the idea. Like you have your license, you have insurance, whatever. So there are 3 checkpoints you need to be familiar with. There's the G1 checkpoint. The G1 checkpoint is, you can see, is the cell growing properly? Is there damage to the DNA? Does the cell have enough nutrients to continue on with the cell cycle? If not, the cell may go into G0. If so, the cell will move into S phase, where S phase is DNA is replicated. Remember, G2 is the preparation. Well, what do you do at the close of your preparation, right before you do something? Let's say you're going on a big trip. You've been preparing for that trip. Right before you leave, you're going to go over that checklist one more time. That is the G2 checkpoint. It's checking for cell size. Is the cell big enough to divide? Has the DNA been replicated? Are there any errors in the DNA? That sort of thing. Is there enough energy for the cell to divide? Cell division requires a lot of energy. And so these are some things that go into that checkpoint. And the third checkpoint is called the metaphase checkpoint. The metaphase checkpoint is very important because this particular checkpoint checks to see if the spindle fibers are attached to the chromosomes at the centromere. Right? If they're not attached, then the cell would go to divide and you'd have a floating chromosome, or you'd have one cell getting both and the other cell getting none, right? And so this particular checkpoint is very important in regulating whether or not cells divide properly. So let's look at the idea of cyclins. Cyclins are another thing that regulate the cell cycle. Cyclins are proteins that regulate the cell cycle, hence the name cyclins. As cyclin concentration builds up in the cell, this cyclin concentration buildup is going to cause cell division. And then after cell division, cyclin concentration completely goes away and begins building up again. Cyclin concentration, or cyclin, is a protein that binds to another protein called cyclin-dependent kinase, or CdK. CdK is an enzyme, right? It is a kinase. Kinase, remember, if you remember, kinase turns something on. So in this case, it turns on the idea of mitosis, right? It turns on the cell dividing. And so as these two things combine, a lot of times what you're going to see, and this is no longer in our course description, but I still teach it because I think it helps students understand. When cyclins and CdK combine, they form something called maturation promotion factor, or MPF. The reason I like talking about it is because it literally tells you what it does. It promotes the maturation of the cell. And so as cyclin and CdK bind together, they're going to build up that maturation promotion factor in the cell until the cell divides, and then there is a point which cyclin production completely falls off, and then CdK, like all enzymes, remains around. Here's another picture that I think is really helpful, and this has MPF in it too. So notice the thick line here has cyclin in it. There are multiple kinds of cyclin. We're going to use the word cyclin generically. Notice CdK levels stay continual the whole time because it's always found in the cell. And as cyclin concentration builds up, maturation promotion factor is going to start building up as well until it gets to one point. Now notice this point is around the end of mitosis. In fact, this point is the G2, or the metaphase anaphase checkpoint. Once the chromosome or chromatids separate, the amount of cyclin begins to degrade and maturation promotion goes away. Right? It makes sense. The cell has done the thing, and now we no longer need to do the thing. In fact, we're going to divide the cell, we're going to chop it in half, and now we're going to have two cells. Cyclin's going to completely go away for a time until that cell begins to, like you can see here, when does cyclin production begin? Begins in S phase, right? Which would make sense. If we're making another copy of the DNA, it's time to start building cyclin. It's time for the cell to start moving toward division. These two proteins are very important in regulating the cell cycle. Let's look at disruptions to the cell cycle. I mentioned earlier, way up here, there's not a picture for it, but if a cell is unable to detect that another cell is next to it, let's say that your paper cut on your finger heals, but yet the cell doesn't know that it is touching another cell. Why would it not know? Well, there's a mutation. Mutation causes those proteins that would normally recognize one another to not recognize one another, and so it continues to send signals to the nucleus to keep dividing. That would have a buildup of tissue. That tissue buildup is typically called a tumor in this case, which tumor is associated with cancer. Cancer is an abnormal cell cycle. It is an unregulated cell cycle, right? So go back up here. What if cyclin never went away? What if the protein that degrades cyclin continued to just produce and had more and more of cyclin? If cyclin was always in the cell, then what would the cell always be doing? It would always be dividing, right? And so this is another example of something that can cause unregulated cell cycle, which is called cancer. The body has mechanisms to fight against this. Cells that the immune system can recognize as cancerous can trigger apoptosis in that cell. The genetic code actually has tumor suppressing genes that cause apoptosis as well. Remember, apoptosis is programmed cell death. But if those DNA are mutated, they're unable to regulate themselves. And so cancer is a multifaceted sort of thing, right? It's not just one problem, but it's a series of problems that come together to create these cells that don't know when to stop dividing. And the body then doesn't know that it should stop helping those cells live. And so that's a real problem with that. So let's look at some practice associated with the regulation of the cell cycle. Mutation in the cell line prevents the degradation of a specific cyclin, resulting in its continued presence throughout the cell cycle. This causes prolonged activity of its partner cyclin-dependent kinases, even when the cell is has unrepaired DNA damage. So basically what this is telling us is this cell is continuing to go through division, even though there's problems with the cell that need to be fixed. Predict the likely outcome of this mutation for an individual cell and its daughter cells. Those cells, those daughter cells, would have the same problems with them, right? And they would also unregulate, and this is going to cause rapid growth. Tumor production, cancer, any of those words would be fine. Explain why the continued activation of CdK is problematic in the terms of cell cycle control. It's because the cell is going to constantly be in that maturation phase. It is always moving toward division, and it's never turned off. That kinase is always activating, and it's always going to continue to turn the cell cycle on. Describe one mechanism by which a healthy cell would normally respond to unrepaired DNA damage. That cell would go through apoptosis, which is called programmed cell death. This was not a difficult FRQ, and so healthy cell, the presence of DNA damage activates checkpoint proteins that arrest us, or it could go into that, right? It could go into G0, could initiate apoptosis. Any of those things would be correct. That's Unit 4. I hope these videos are helpful. If you have any questions at all, please do not hesitate to leave me a comment, and I will answer all my comments. Thanks for watching.